here. Looks like the button has been pressed for recording. Thank you. So yeah, it's a pleasure to be here. I think this is a great idea. I think of some of the things the positive things that have come out of the pandemic. I think being able to connect virtually like this across countries and boundaries and time zones and to hear speakers that you would normally not hear is a great idea, and I hope very much as a community we can continue this forward once we get back to normal. We can still have these virtual activities where we can bring everybody together. So long may it continue, and thanks to the ESMTB for initiating this, and it's a pleasure and an honor to be the first speaker in this series. So as Angelique has very nicely said, I've been working on cancer modeling since my PhD, and gradually being able to look at lots of aspects of cancer modeling. And this is very recent work, and it's the reason that I became interested in it was that I was interested in it for a long time, but never found the actual time to get to look at it, because metastatic spread is a very important aspect of cancer growth, and as we'll see, it's the thing that really causes most deaths. And recently, a few years ago, I had a PhD student who was jointly supervised by Tommaso Lorenzi. I think Tommaso is there with us today as well, and she wanted to look at metastatic spread, so we initiated this project with her. So the talk will just give a little overview of cancer. I'm sure everybody's aware of what cancer is, but a little overview of what cancer is, a disease, and then focus on initially some invasion modeling, so how cancer spreads locally in the tissue that it's in, because that's very closely related to how metastatic spread, which is the spread to secondary organs, occurs. So before looking at the metastatic spread, we'll go back and I'll sort of revisit some work I did some time ago, which has led ultimately to this new modeling of the metastatic spread, which is very recent work. And then I'll mention towards the end some very recent developments that we've taken the metastatic modeling forward in slightly different directions. So about 20 years ago, there's a famous paper that's published by two people, Hanahan and Weinberg, and it was called The Hallmarks of Cancer. And it's now had tens of thousands of citations, and anybody who works in the field, either cancer per se as experimentally oncology or modeling, knows this paper simply as Hanahan and Weinberg. And so if you talk about Hanahan and Weinberg, everybody knows what paper you are talking about. And in their original paper published in 2000, called The Hallmarks of Cancer, the Hanahan and Weinberg focused in on differences between cancerous tissue and normal tissue, and cancerous cells and normal cells. And they tried to get a sort of minimum set of things that they felt made cancer different from normal tissue and normal cells. And they came across 6 key points, which they called hallmarks. And these are known as the hallmarks of cancer. In a more recent paper in 2011, they updated things a little bit, but the basic hallmarks remain the same. And there are 4 hallmarks that are related to the cell or the subcellular activity. One is that cancer cells have excessive proliferative ability, and so that gives them a proliferative advantage over normal cells, and that's why you get a mass of cells forming in the first place. Also, cancer cells, through various pathways signaling pathways that have gone wrong, have the ability to resist cell death, so they don't undergo apoptosis, which is the normal mechanism of cell death and growth control. They have mechanisms of being able to keep proliferating even in the absence of limited nutrient. They have the ability to switch from oxygen-driven nutrient to glucose and use a glycolytic pathway, which under normal circumstances is very inefficient, but they can switch to that, and then that helps them to keep alive and keep proliferating. And then they have the ability to escape or evade the normal mechanisms that the body tries to use to suppress any abnormal growth. So these hallmarks, these differences are at the cell or subcellular level, and I won't talk about any of these today really. But the two that allow cancer to become a very dangerous disease are, first of all, is, and again, these are also linked, the ability to induce blood vessel growth, which is angiogenesis or tumor-induced angiogenesis. And so Angelique has also done very nice work recently on modeling angiogenesis and the blood flow and the delivery of oxygen and nutrients to the tumor. And that's also linked with the other, that's a cellular or a tissue level activity. And the other thing that makes a cancer different from normal tissue and difficult to treat is that the cancer spreads locally into the tissue, and then combined with the blood vessels, if a cancer cell gets into a blood vessel and then it gets into the main circulatory system, the cancer cells are transported throughout the body and can eventually get out of the blood vessel system into a secondary organ. So metastatic spread means there are secondary tumors which are not in the primary location, but they've derived from the primary tumor. And that is what makes cancer really very difficult to treat. Once you have metastatic spread of the primary to secondary locations in the body, the prognosis for a patient is not very good at all. So if we could stop metastatic spread, far, far fewer people would die of cancer. And this is a statement from the World Health Organization. This is from its webpage. This just gives an overview, because cancer is a global disease. Unfortunately, many millions of people a year die from cancer globally. And this is the World Health Organization description of cancer. It's a generic term for a large group of diseases that can affect any part of the body, because any cell can go cancerous and develop into a cancer of wherever the cell comes from. And then a defining feature is the rapid creation of abnormal cells. That's the excessive proliferation, which can invade locally into the tissue and then spread to other organs, and that's the metastatic part. And then the final sentence is the key here. Metastases are a major cause of death from cancer. So it's the spread that really complicates treatment and makes it difficult to cure. And this other statement here is taken from the original paper of Hanahan and Weinberg in 2000, where they say that these distant settlements of tumor cells, the metastases, are the cause of 90% of human cancer deaths. So again, if we could stop, just looking at these figures, if we can stop metastatic spread, we'd stop a lot of deaths from cancer, which would certainly be a good thing. So trying to develop a model of metastatic spread, we were interested just to try and help people make better decisions or inform clinicians as to when a primary tumor may have metastasized, because then that allows them to perhaps act more quickly or bring in different treatments to try and help at the earliest possible stage, intervene at the earliest possible stage for patients. So that's the long-term goal of this modeling. And just a little overview of solid tumor growth, because that's what we're going to focus on. Of course, tumors, cancers of the blood, leukemias, they are of a different nature and require different types of modeling. So for solid tumors pass through an early growth phase called an avascular growth phase. And that's because nutrients are delivered to the tumor cells via diffusion of nutrients alone, normally oxygen. And what you get developing is a sort of spherical mass of cells. And in the laboratory, when you grow these in vitro, these are called multicellular spheroids. And you can see here, here's a picture of a multicellular spheroid. And this grows to a size of a few millimeters in diameter with about 1 million cells. And so it's limited because the nutrients only diffuse from the surrounding tissue. And basically, there's a balance between cell birth and cell death. As the oxygen diffuses into the tumor, you can see from this figure B that this is with some staining. The dark cells are live proliferative cells, and the cells in the middle that are lighter, these are necrotic cells, and they've died because the level of oxygen in the center of the tumor has fallen below a viable threshold, and so they're necrotic. And so there's a balance between cell birth and cell death, and this reaches a sort of steady state level at about a few millimeters in diameter. And at that stage, there's a third region when you look at the cross-section of the tumor. There's a middle region, which are composed of quiescent cells. So they're neither dead nor alive. They're in this middle zone that if the oxygen levels improve, they would become proliferating and start proliferating again. And if the oxygen level worsens, they would become necrotic. So there's a three-phase structure in the interior of these spheroids, composed of proliferating cells on the outside, and there's quite a thin rim of them, just a few cell layers deep. And then there's a middle zone of quiescent cells, and then there's a final inner zone of dead or necrotic cells. And that is in some sort of dynamic equilibrium at a few millimeters in diameter. And if that's all that a cancer did and grew to a few millimeters in diameter, almost nobody would die. But of course, that's not what happens. And angiogenesis takes hold, and I don't want to go into angiogenesis. That would take a few seminars on its own to describe all the interesting things that happen in tumor-induced angiogenesis. But basically, blood vessels grow towards the tumor. That brings oxygen, and it can grow because it's got more oxygen. But in addition to the tumor-induced angiogenesis, cancer cells also have the ability to start invading the local tissue through secretion of enzymes. And here are a couple of pictures from a pathology textbook where you can see that the initial cancer, this is an epithelial tissue, so this is a carcinoma. You can see that rather than remain localized in the epithelium, it's now broken through and degraded the basement membrane and is now invading in this sort of irregular way. And this is another image from a pathology textbook where this is started out as a solid mass, a spherical mass, and now you can see through local invasion, you get these projections of cancer cells going into the local tissue and spreading outwards locally. And that's caused by the secretion of enzymes, which we'll see in a few slides later. And the cancer was known to the ancient Greeks. The word carcinoma derives from the Greek for a crab. The ancient Greeks could see that some people who died from cancer had these lesions inside them that looked like a body of a crab with the legs of the crab spreading into the tissue. And the word cancer itself is also the Latin for a crab. So the ancients knew of the disease many thousands of years ago. And unfortunately, they were unable to do anything other than record it, so to speak. And here's a more up-to-date image. This is from a relatively recent paper from a group in Japan who are trying to classify the invasion of, in this case, this is cancer of the esophagus, your tube from your neck to your stomach. So they looked at the patterns of invading cancer cells in the esophagus and tried to connect that with the prognosis for patients. And they came up with three classifications. There was they called it an invasion pattern A. And in this case, you can really distinguish the cancer tissue from the normal tissue. This is with staining. So this darker purple is the cancer. This is the boundary, the edge, the border between the cancer. And below here is the normal tissue. So you can quite clearly distinguish between what's cancer and what's normal. And that has a good, if you find the tumor, find the cancer at this stage and treat it, the patient has a good prognosis for survival. And then they have invasive pattern B, which is a little more heterogeneous. The cancer cells, it's not so uniform. You can still see that there is a sort of boundary between the cancerous tissue, which is darker purple, and the normal cells, which are lighter purple. The boundary is here, but it's a little more irregular. That has not such a good prognosis. And then finally, they classified what they call an invasive pattern C, which is very heterogeneous, and the cancer tissue and the normal tissue are completely mixed together, and it's very difficult to see where the boundary is. There's certainly cancer cells, darker purple down here, but there's also normal tissue up here. So just surgery is still a treatment method. And so where you, in case A, you could quite clearly, if you're a surgeon, just cut here and you would be confident that you would remove the cancer. If you're presented with pattern C, it's not clear where you want to cut away the cancerous tissue because it's mixed in with the normal tissue. And so there's a bit of a dilemma then that how much tissue do you cut out? Because if you cut away too much healthy tissue, that can also damage the patient in other ways. So when it gets very aggressive and invasive, it's difficult to treat via surgical techniques. And this invasion pattern C has a very poor prognosis. If it's discovered at this stage, it's quite advanced, and survival is not nearly as good as it is in invasive pattern A. And so just to summarize the key biological mechanisms of cancer invasion, there are three main components. There's the cancer cells themselves, and how they interact with the extracellular matrix. That happens through a class of enzymes called matrix-degrading enzymes. And there are two main classes. There are matrix metalloproteinases, MMPs, and there's also the urokinase plasminogen activator system. And so these are classes of enzymes. They're secreted by the cancer cells and they degrade the tissue. They operate in slightly different ways. This talk will focus on MMP interaction with the matrix, but the urokinase plasminogen activator system is also very interesting. In fact, I think I just saw George Lolis come along. George developed a very nice model some years ago looking at the urokinase plasminogen activator system and got some very interesting results with that invasion system. So this talk will focus on MMPs, matrix-degrading enzymes. They are secreted by the cancer cells. They degrade the tissue or with components such as collagen and fibronectin, which are adhesive components for the cancer cells. And so that allows the cancer cells to either proliferate into gaps or use the matrix components that are exposed by the matrix-degrading enzymes in order to adhere and move actively into the local tissue and spread. And so that's what produces those classic spreading pictures that we saw a few slides previously. So if you're going to develop a model of cancer invasion should include cancer cells, extracellular matrix, and the matrix-degrading enzymes at a very minimum. And the key mechanism of migration of the cancer cells that is to be included is a mechanism of haptotaxis. So that's interaction with the matrix through adhesion. And this is a nice experiment that was carried out with some cancer cells. And these cells have been put on a glass slide, and they can't adhere to the glass slide, so they remain localized and cannot spread. If you put some component of the extracellular matrix on the glass slide, the cells can interact and adhere, and you can see now that they've started to spread. And then if you put a component of the matrix on that they get a very nice adherence to, in this case, it's a component called tenacin, which is a protein like collagen or fibronectin, you can see the spread is very wide indeed. So interaction with adhesive components allows the cancer cells to spread, and that you move up adhesion gradients or matrix density gradients, and that process is called haptotaxis, which is very similar to chemotaxis, but the underlying mechanism that the cells are responding to doesn't diffuse. In chemotaxis, you have a diffusible chemical that sets up a gradient. In haptotaxis, it's the gradients in the matrix which are caused by the degrading enzymes. So it's similar mathematically, but biologically, the haptotaxis involves no diffusion of the underlying chemical, in this case, the matrix. So if you want to develop a sort of high-level, let's say, macro-scale model of invasion using partial differential equations, you'll have an equation for the cancer cells, an equation for the extracellular matrix, and an equation for the matrix-degrading enzymes. If you want to distinguish between matrix-degrading enzymes or different components of the matrix, you can add in some more equations, but we can focus in on just a basic core model with the cancer cells. And the cancer cells produce matrix-degrading enzymes at some rate, and the matrix-degrading enzymes M, they diffuse and decay, and then the matrix-degrading enzymes, when they interact with the matrix, they degrade the matrix at some rate gamma. You can put in a term which models remodeling, so the matrix like to try and get back to a healthy level if there's fibroblasts brought in to repair the matrix. You can do that in competition with space competition with the cancer cells. And then the cancer cells themselves, you can include proliferation. This term includes space competition with the matrix. And then here we have, again, little bit of random motility for the cancer cells, which is probably always there. But the main mechanism of migration is the directed migration, the haptotaxis, which is motion up density gradients of the extracellular matrix V. So the system is coupled through the cancer cells secrete the enzymes, degrade the tissue, which forms gradients, and then the cancer cells use those gradients to move. And you can make this model even simpler, which is what we did 20 years ago now. In the first instance, if you ignore the proliferation of cancer cells, if you look on a relatively short time frame and just look at the migration of the cancer cells rather than the proliferation, we'll see how we add proliferation in later. So if we take away the proliferation of the cancer cells and the matrix remodeling, then we have a very, very simple system for these three equations. And this was published, as I say, 20 years ago with a colleague I worked with then in Dundee, Sandy Anderson, who's now leading the Institute for Maths Oncology at Moffitt Research Cancer Institute in Florida. And we were working with some clinicians, Professor Bob Steele and Professor Alistair Thompson. Professor Steele was an expert in the local hospital, in the teaching hospital related to the university on bowel cancer. And Alistair Thompson was and still is an expert in breast cancer. And both Bob and Alistair were interested in the problem I alluded to a few slides ago, whereby when they were operating on a patient with advanced cancer, breast or bowel, and they wanted to remove it surgically, they wanted to work out how much tissue to take away in order to be guaranteed that the cancer wouldn't recur in a few years' time. Because if you leave a few cancer cells in the external tissue, then that allows the possibility of the cancer to come back in a few years' time. The cancer cells have the ability to proliferate and survive under harsh conditions. And certainly in the breast cancer, the data from surgery in the breast cancer domain showed that although a lot of the surgery was very successful, a proportion of women came back 4 to 5 years later with the same breast cancer in the same location. And that was probably because some cells had been left in the first operation. So they were trying to optimize their surgical techniques to try and remove as much tissue as they could to guarantee that all the cancer had been taken while not damaging too much of the breast or going for radical surgery, which has other complications. So I became interested in the invasion problem from that aspect. So from a purely PDE point of view, if you look at that system and solve it as a system of PDEs numerically and look at the simulations, and this is just some nice one-dimensional simulations to get a flavor for what the solutions look like numerically. And of course, it's a reaction diffusion taxis system, so you would expect some kind of traveling wave-like behavior. And in fact, obviously, if you put in the logistic term, you're just going to get like a normal Fisher-type traveling wave, logistic Fisher wave. But when we took away the proliferation for a certain parameter set, we noticed the following interesting result. This is the domain, the one-dimensional domain. And so we have a cancer, a solid tumor in the left-hand part of the domain initially, with extracellular matrix and with the enzymes. The enzymes are diffusing and degrading, and so that sets up the gradient of the extracellular matrix. And then via haptotaxis, the cancer cells invade the matrix while degrading it. And you can see that what's happened here is that a component of the tumor has broken away. The main body of the tumor is here because it's the matrix has been degraded too much. And so at a later stage, this is a sort of a one-dimensional interpretation of those penetrating fingers or columns of cells that are invading into the local tissue. And if you then simulate that in 2 dimensions, you get a more qualitative feel for this type of invasion, this heterogeneous invasion. These are the simulations in 2 dimensions, and you can see that there's a heterogeneous spread. This is partly down to the strong haptotaxis effect, and also we made the matrix itself vary in density, so the matrix is not smooth, homogeneous density. There are natural variations in the density, but you can see that qualitatively you get a heterogeneous spread, and components of the tumor have sort of broken away from the main body, and that qualitatively models what we were seeing in the pathology textbooks, with these heterogeneous columns of fingers spreading into the local tissue. But at the surgical level, it only takes a very small number of cells in order to be left for the tumor to recur. And so this PDE model is at the level of cell density, so we're still talking about a lot of cells in order to have a viable PDE with lots of tens of thousands of cells, etc. So we wanted to try and develop a technique which allowed us to track smaller numbers of cancer cells, which would be more accurate and realistic for the surgeons. So in order to do that, we developed a technique that we used to model tumor-induced angiogenesis, where we were looking at the development of blood vessel networks, which are discrete connected blood vessels. And so to do that, we go back to our original system, and the equation for the cancer cells, you simply discretize that with some very, if you're a numerical analyst, you would never do this, but you would just explicitly discretize it with some centered finite differences and an explicit time step. You can always guarantee that stable in positive solutions. If given a space step delta x, you choose a time step delta t small enough, that works in a very crude sense, but it's very numerically inefficient, but it works very nicely for this technique that I'm about to explain to you. So you discretize that in a normal way, and if it's a two-dimensional domain, you're working on a 5-point stencil. And so when you do that, the discretized equation for the cancer cells looks like this. So this discretized equation says that the cancer density, if you're solving this numerically as a density, it would say that the cancer cell density at grid point ij at the next time point n+1 is related to the cancer densities on the 4 neighboring grid points, I-1, I+1, so it's to the left and right, J-1 and J+1, that's up and down, and then at the grid point itself at the previous time step n. And then you have 5 coefficients, P0 up to P4, and that's related to the discretization of the diffusion operator and the discretization of the haptotaxis term. And if you look at the 4 terms for the neighboring grid points, as you can see here, there's a component down to diffusion and a component down to the discretization of the haptotaxis operator. And so if you have gradients in your system, you introduce biases. And so this, if you interpret these coefficients P0 to P3 and P4 as the probabilities of movement, then you can generate the movement of an individual on the grids rather than solve for the density at the next time step. And that's what we'd done just a few years earlier to model tumor-induced angiogenesis and the movement of individual endothelial cells responding to tumor angiogenesis factor, VEGF, where you can track the motion of individual cells which make up individual capillary elements. Then if you do that at each time step, if you have individual cells at grid points, you can move them from one grid point to the next, depending on the size of these coefficients, P0 up to P4. And if there are any gradients in the system, then some of the coefficients are bigger than the others. And so if you generate a probability of movement each time step, you're more likely to choose those coefficients. And so you're more likely to move in the gradient of the higher concentration, which is where the gradients are coming from. So in effect, what this technique does is it generates a very simple way to generate a biased random walk of individuals. And in this case, the individuals are cancer cells that are invading the tissue. So this is exactly what we did. We took the discretized equation for the cancer cells, and so our discrete equation looks like this. And so we were able to move the cells from grid point to grid point at each time step of the simulation. And what we were then able to do was add back in proliferation at this discrete level. So rather than put the proliferation in the PDE as a logistic term, we are now able to allow each cancer cell to proliferate every 24 hours, which is roughly what an invasive, aggressive cancer does. So every 24 hours, we take our cancer cells and we divide them into 2 daughter cells, and then the 2 daughter cells can move on the grid points driven by the movement probabilities. The other thing that one can do when you have a discrete, so we call this a hybrid system because we have discrete cancer cells responding to matrix density and matrix degrading enzymes, which are still both PDEs. So in addition to cellular proliferation, you can model cell adhesion in a phenomenological way. You can allow 2 cancer cells to adhere to each other with some adhesion strength. And over time, you can allow that adhesion strength to weaken, because that's what happens. Cancer cells lose adhesion as they become more aggressive. You can allow the cancer cells to mutate, and with each mutation, they become a little more aggressive, so they can proliferate more quickly. They can have a bigger haptotaxis coefficient, so they interact with the matrix more strongly, and that allows them to move more rapidly into the matrix, into the extracellular matrix, away from a sort of normal average cancer cell. The more aggressive ones that have mutated can spread more quickly. So this hybrid technique combines, our idea was that it combined the best aspects of PDE modeling with the best aspects of individual-based modeling or with cellular automaton-like rules. So you put the best of both together and you generate this hybrid model. And in doing that, we were then able to simulate cancer invasion at an individual level. And so these are the simulation results in 2 dimensions, in a 2-dimensional domain with some individual cancer cells placed in the center of the domain. And then over time, they spread as individuals interacting and proliferating, interacting with the matrix, haptotaxis, and proliferating. And then at a later stage of the simulation, you can see that they've spread out and there's a large mass of cells, but there are a number of cells that are a reasonable distance away from the central mass of cells. And for example, here there's a cell that's moved away quite far. And so the idea was that this would represent what would be a visible margin. This is easy, if this was a surgeon, it would be easy to see most of the cells there because they're very dense, but we could then estimate how far into the tissue that you would have to cut in order to be assured of getting all the cancer cells surgically removed, and that would maximize the chance of the cancer not recurring in the future. And when we first showed our surgical colleagues these simulation results, they thought that we'd managed to get them from some medical journal because this is exactly what they saw, certainly in the breast cancer. This is exactly the image that they saw with a mass of cells that were visible and then cells that were penetrated into healthy tissue. So our idea at the time was to try and develop this further to get more data, to make the model more accurate, and eventually to produce a tool which would inform surgeons of what they had to, how much tissue they had to surgically remove in order to maximize the chance of the patient survival with non-recurrence of the cancer at a later time. Unfortunately, our colleagues didn't have enough time to devote to the research aspect of their work at the time, and so the modeling was left there. And we had to move on to other projects. So we had a nice invasion model that required more work to take it further. And that's basically where it was for about 15 years or so, until I had a PhD student at St Andrews, Linnea Franson, who's jointly supervised by Tommaso Lorenzi. And Linnea was interested in cancer invasion and metastatic spread. So in order to set Linnea off on her PhD work, I asked her to go back and look at what had been known about invasion, cancer invasion from a biological oncology point of view in the intervening 15 years, in order to develop the initial invasion model before we would then move on to the metastatic spread model. So 2 main points Linnea found out, had 2 main discoveries, so to speak, had been focused in on in the biology side of cancer. The first was that there was a lot more known about what happens to the invading cancer cells. And a very important aspect of invasion, which is now well known these days, is something called EMT, which stands for the epithelial mesenchymal transition. So invading cancer cells from over 90% of tumors are carcinoma, which develop from epithelial tissue. So epithelial cells actually, in order to invade, because epithelial cells are rather non-motile, they have to change phenotype into mesenchymal cells, which are more motile. So there's been a lot of work on this phenotypic transition from an epithelial-like phenotype to mesenchymal phenotype over the years. And so that's led to a lot more knowledge of this epithelial mesenchymal transition. So that's something that should be included in an updated invasion model. The other aspect of invasion, which a lot more work has been done on, is the enzymes themselves. And it's now known there is another class of enzymes in addition to MMPs and UPA. So they're both diffusible and diffuse into the matrix. But there's another class called membrane-bound MMPs. So they're secreted and then are rebound to receptors on the cancer cell surface. So that was another aspect of the invasion model that had to be improved upon. And so this is a little schematic of the epithelial mesenchymal transition epithelial mesenchymal transition. So here are some epithelial cells, which normally are stuck together with intercellular adhesion bonds. And then over time, they change phenotype into mesenchymal cells, which are more invasive and more motile. And there are key differences between the two cell types. Mesenchymal cells are more motile, and they're the ones that are actually leading the invasion. The other aspect of mesenchymal cells, which is very important, is that they can interact with blood vessels and enter the blood vessels, and that process is known as intravasation. And it's the mesenchymal cells that are actually secreting all the enzymes. The epithelial cells, the epithelial-like phenotypes, they don't express any enzymes. And although they can invade, they have to have the presence of the mesenchymal cells. So they sort of play follow my leader, if you like. So mesenchymal cells are motile, so epithelial cells can link on and adhere and form little clusters, but on their own, they can't do any invasion. However, the epithelial cells are more proliferative. So the mesenchymal cells are motile, and the epithelial cells are proliferative. So you've got both aspects of proliferation and migration in these two phenotypes. And that's led to models of cancer invasion over the years at a PDE level of go and grow or proliferate and migrate. So you have two classes of cells, one which proliferates only and one which migrates only. And you can have a switch between the two populations, and that's led to some interesting models, and you can do some traveling wave type analysis. But we want to embed this at the hybrid level of individual cells. And then the other aspect, which is very important, these membrane-bound enzymes, these were discovered in the lab of Stephen Weiss in 2009, and this is from the abstract from that paper there. And this very nice work, showed that cancer cells have an absolute requirement for these membrane-anchored metalloproteinases. They're called MT1 MMPs. An absolute requirement for invasion. So unless these membrane-bound proteins, matrix-secreting enzymes are present, invasion is much less reduced or doesn't happen at all. And Stephen Weiss and his group showed this in a very elegant experiment. They took some cancer cell spheroids and embedded them in some collagen gel assays, and then they they had a control experiment. They had an experiment which knocked out the diffusible MMPs, and then they had an experiment which knocked out the membrane-bound MMPs. And so they looked at the three results of these experiments. And as I say, they embedded some cancer cells in collagen gels, left it for 3 days, and then came back and looked at how invasive the cancer cells had been. And these are the results. On the left column here is the control experiment. So initially the cancer cells are embedded in the collagen gel, and then 3 days later you can see they've spread in a standard sort of invasion spread pattern. In the middle column is what happens when you knock out the diffusible MMPs, and there's almost no difference between this and the control experiment. And then the right-hand column is what happens when you knock out the membrane-bound MMPs. And when you knock out the membrane-bound, the invasive spread is very, very much reduced, and it almost doesn't invade at all. So their conclusion was that without these membrane-bound proteinases, invasion was really hard to take place. Now, there are some nuances there. The diffusible enzymes, actually, they need activated to in order to work. So this is an in vitro experiment. So in vivo, perhaps slightly different, but this experiment does show the real importance of the membrane-bound MMPs. So the first step of our metastatic spread model was to update the invasion model. And so we now have two cancer cell phenotypes. We have an epithelial phenotype and a mesenchymal phenotype. And the mesenchymal phenotype, they're the ones that are secreting the enzymes. So these are the diffusible enzymes. And we now have, we still have cancer cells, diffusible enzymes, and matrix. Rather than get into a very complex model with a multi-scale model with receptors and enzymes, and we decided simply to model the membrane-bound effect, membrane-bound proteinases, through a simple interaction term with the mesenchymal cancer cells in the matrix. So when mesenchymal cancer cells interact with the matrix, there's some level of degradation, which is more or less what happens, because the membrane-bound MMPs are on the surface of the cancer cells, so it's as if the cancer cells themselves are degrading the extracellular matrix. So that's the updated invasion system. Once again, we discretize both cancer cell phenotypes, epithelial phenotype and mesenchymal phenotype, and then we add in at the discrete cell level the proliferation, migration through haptotaxis with haptotaxis coefficients, adhesion. Of course, for the epithelial cells, they'll be far more proliferative than the mesenchymal cells. The mesenchymal cells will be more haptotaxis than the epithelial cells. And also there'll be a transition between the two. There's a probability that epithelial cells can become mesenchymal. So we can do all that at the individual base level with a modification of the cellular automaton rules that we have. And in order to take our invasion model a step further, we first of all benchmarked our parameter set against the model of Weiss et al. So these are our simulations. We took our parameter values to try and connect with their nice experiment. So this is on correct spatial scale and temporal scale. And so this is a control experiment with everything. And then we removed our diffusible enzymes and we tuned the parameters in order to have the spread in a very similar way to the control experiment. And then when you knocked out the membrane-bound enzymes, again, we have a very small spread compared with the control experiment. So we've benchmarked our parameter set against the Weiss model, and that's all we did in terms of parameter connection. So that was what we felt an improved invasion model. And so then we need to take that invasion model and connect with a metastatic spread. And so just an overview of the metastatic, it's called the invasion metastasis cascade. So here's our primary tumor, and that begins to invade through the mechanisms that I've just described. And then these are the mesenchymal cells. They can invade as individuals, but you can also have collective invasion with clumps of epithelial cells connected to mesenchymal cells. And if there are any blood vessels nearby, then the mesenchymal cells can get into the blood vessels through intravasation. There's experimental data giving probabilities of intravasation success. Or you can get these clusters of mesenchymal epithelial cells intravasating, but they're led by the mesenchymal cells. And then once you're in a blood vessel, the cells can either circulate as individuals. So these are called CTCs, circulating tumor cells. And again, there's a lot of data there because people know that there's a lot of tumor cells. If you have a primary tumor, they get into the blood vessel system and there's a lot of CTCs, but only a few actually succeed in becoming metastasizing to distant organs. So there's a probability of survival of the tumor cells in the blood system, and then at a secondary site there's a probability of extravasation, and then once you extravasate into the new tissue, you then grow again and invade as per the original invasion model. So our metastatic spread is based around this new invasion model, but we have now developed what we've called a multi-grid framework. So rather than just have a primary grid where we look at the invasive spread and see the patterns of invasion, we allow the cells to move from the primary grid to a secondary grid, representing a secondary organ. And I'll just explain that in a few slides' time, but that's the core of the metastatic model. So we have multiple grids with a primary tumor on a primary grid, and then secondary tumors can move to secondary grids. And for the purposes of the work that we did, we connected with a very nice piece of work in the United States from breast cancer patients. And so we have a primary grid which represents breast cancer, and then secondary grids representing the lungs, the bone, and the liver. And that was all driven by this nice study from 2013 in United States in Texas. And they studied over 4,000 breast cancer patients and looked at where the breast cancer spread and what was the sort of favored secondary sites. And the three top sites were bone, liver, and lung. And so we focused our model on a secondary spread to three distant organs: bone, liver, and lung. You can put more in, it just takes more simulation time. The other aspect of the metastatic cascade we need to account for is the interactions with the blood vessels. So we need blood vessels in our system, and then the probabilities of intra and extravasation, the probability of survival in the blood, and then getting out into the secondary tumor. So this is our primary grid with our epithelial mesenchymal cancer cells in the middle, very similar to before, but now on random grid points we've placed blood vessels. So you can think of this as a two-dimensional cross-section of the three-dimensional tissue. And so what we're going to do is allow our epithelial mesenchymal cells to spread through our invasion model. And if they come in encounter with the blood vessels, there's a probability of intravasation. And it'll depend if you're a mesenchymal cell or an epithelial cell or a cluster. You can also include things like ruptured blood vessels, because the interactions with damaged blood vessels is slightly different to the interactions with normal blood vessels, and that can also be accounted for. And so here is our little flowchart schematic diagram. We have invasion on our primary grid. If you encounter a blood vessel, there's a probability of intravasation. If you're successful in intravasation, there's a probability that you survive. This is data from the literature. So you either, once you're in the vasculature, you either die or you survive. And then if you survive, there's a probability you'll go to one of the three secondary grids. Again, that's driven by the probabilities from the study in Texas on the breast cancer patients. So just very briefly, the intravasation, all these are just done by probabilities. If a cell encounters a blood vessel, there's a probability that it successfully intravasates, and that depends on the type of blood vessel you encounter. You can either do that as a single mesenchymal cell or as a cluster of epithelial cells with mesenchymal cells. When you're in the vasculature, there's a probability of survival. And again, that will depend on whether you're a cluster of cells or a single cell. And that's all been obtained from the data in the literature. And then there's a probability that you extravasate at one of the secondary sites. And then once you get to the secondary site, you just grow and invade as if you were at the primary site. And so this flow chart here captures this, is our little algorithm for this invasive metastatic cascade. So if I just move now to the basic simulation results, these are our, this is our primary tumor, this is our breast cancer. So here's our epithelial cells at the beginning and mesenchymal cells, and then they spread, grow, proliferate, migrate, invade. And you can see that at a late time, there's mixtures of both. the epithelial cells, there's a lot of them because they've proliferated, but the mesenchymal cells are at the margins. Okay, they've migrated further into the tissue, and when you combine them both, it's just a central mass. It's just a massive mixture of mesenchymal and epithelial cells, but the mesenchymal cells are largely on the outside, really. And then as they've been invading, they've been encountering blood vessels, and so there's a chance that they've metastasized, and then that can be done by looking at the secondary grids. At an early time point, we can see that there's already been a spread to the lungs. And then at a later time point, we've got multiple spread to the lungs. We've got multiple sites, and that's both epithelial cells have succeeded, and at a later time, the mesenchymal cells have succeeded. And then in the bones, at an early stage, you don't have much spread to the bones, but at a later stage, you can see that both epithelial and mesenchymal cells have managed to get into the bones, and qualitatively, that's what's observed. And then finally, for the liver at an early stage, not much happens, but at a later stage, we're seeing, you maybe just see down here, that some mesenchymal cells have managed to get out. And if we run the simulation for longer, then we see both mesenchymal and epithelial cells get into the liver and set up secondary liver tumors. And so qualitatively, that's exactly what's observed. And of course, that's all driven by the probabilities that are in place, but the model is indeed capturing metastatic spread to these secondary organs via this multi-grid technique. So just an overview of the results, that we have an improved invasion model that connected very nicely with some invasion work by a Finnish group. We had good correlation with invasive speed and invasive spread and patterns of invasion. And then the secondary spread connected qualitatively with the experimental, with the results that had been observed from the study in the United States. So we now have a framework for modeling spread from a primary to a secondary tumor. And that's as I say, the title of the talk was a framework. And that's what we're hoping to develop further in the coming years. We need to focus in on a few places. We need to do some further model development, some refinement of the modeling, parameter sensitivity testing, etc. Again, as we were 20 years ago, we'd like to connect with real data and ultimately help surgeons and give data to surgeons to make sort of more informed decisions to try and make optimal clinical decisions for patients. We've got this hybrid framework. We'd like to take the modeling in a direction of connecting to our angiogenesis model in a more explicit way. At the moment, it's just if you're on a grid point with a blood vessel, it's a probability of transport. But we'd like to take our tumor-induced angiogenesis model and couple that and do a more interactive model and a more accurate model there. And then also, once a tumor cell is in the blood vessel, again, the transport survival is just driven by a probability, but we have a flow model as well. So once a cell gets in, we'd like to look at how the flow properties of the blood in these microvasculatures, how they affect the cancer cells, and try and get the survival driven by things like shear stress. And so again, a more accurate description of survival rather than just a probability, which is got from experimental data. So just coming to the end of the talk, I'd just like to point out this was published a few years ago now in the Bulletin of Mathematical Biology, and it was Linnea who did all the hard work and helped by Tommaso, and also a former PhD student, Andrew Burgess. There's an interesting little connection here with, I think, the flexibility of mathematical modeling. Andrew Burgess worked with me in Dundee on a PhD project on host parasitoid systems with the local Crop Research Institute. And Andrew was interested in looking at how pests on crops could move from field to field. And so some of the ideas that Andrew developed in his PhD for ecological modeling, that's what underpins this multi-grid system for the metastatic spread. So there is a nice connection from ecological modeling that I did some time ago with Andrew to the modeling that we have in this new metastatic framework. And then last year, Linnea updated the model a little bit more, because in addition to epithelial mesenchymal transition, EMT, there's also the reverse. Mesenchymal cells can also revert back to a more epithelial-like phenotype. So there's something called MET. So you get EMT and MET. And so we have already incorporated that into the model, and that was published last year. And this has also led to some really new thinking about the hybrid technique. Because at the moment, what's been described here is either a PDE model, which is very macroscale for cell density, or a discretized model which tracks individual cells. But of course, reality is somewhere in between. You get small clumps of tumor cells, not just individuals. And so this has led to the development of a new hybrid continuum discrete model. And we started applying that to an organotypic assay to try and connect with the data. And the very nice work on the new hybrid technique was led by a current colleague in St Andrews, Nikos Svakianakis, whom I'm sure many of you already know. And so Nikos has developed the hybrid model with a nice stochastic transition technique to move from the PDE to the discrete system. So it's a more sophisticated way to do it than just simply discretizing the system. And that appeared last year also. And then very recently, we've applied this to look at invasion in organotypic assays, because the data there is very nice and you can really visualize the invasive spread. Obviously in patients, you're seeing things at a later stage. In an in vitro assay where you have organotypic gels and you put some cancer cells on the top, you get a really nice visualization of the spread of the tumor cells, the cancer cells into the assay. These are three-dimensional assays, and then they have a cross-section and you examine how the cancer cells have invaded. And so this is from a paper. These are some cancer cells being plated on the top of a collagen gel, and over time the cancer cells invade, and as you can see, they're rather dense at the top, and the little clumps of cells have broken off and they've invaded in a sort of heterogeneous way. This is this experimental images of the cells at the beginning of the assay, and then a few days later they've invaded in this heterogeneous way. And so this is some simulation results of some very recent work. This is our new hybrid invasion model. This is our connection to these organotypic data. You can see these are our cells at the beginning of the, very early of the stage of the invasion, and then at a later stage, about 2 weeks into the invasion, we have a nice qualitative match between the invasion pattern that we're seeing in our simulations and the invasion pattern that we're seeing in the in vitro data. And this is all done in a nice three-dimensional framework. And so we've simulated this in 3 dimensions and then taken a 2D cross-section, just as exactly as happening in the in vitro experiments. And so I'll just finish to say that this is published online literally in the last few weeks, the Journal of Theoretical Biology, and all the very nice work was done in combination with Linnea, and Nikos developed this in a very nice way and connected with the data as well. So I'll just finish here by saying thanks to Linnea and Nikos for all their efforts and all the work over the last 5 years. And I'd like to thank you all for your attention this morning, this afternoon, and I'm very happy to take any questions. Thank you.
Thank you.